Well, good afternoon, everybody, and thank you so much for joining us today. And very, very warm welcome to Dr. Pedro Bergamo. Thank you, Pedro, for joining us and for being willing to do this presentation today. We're really looking forward to it. And I'm going to hand over to Anina now, who will introduce Pedro more formally, and then we'll go ahead with the presentation. And Nina will be chairing the session. So thanks once again and welcome everybody.
Thank you, Glynis. Good afternoon, everybody. Thank you so much for being here. So we've had 3 SAAB webinars this year so far by our 3 medalist winners. And today we've got an exciting seminar on pollination ecology. And so today we welcome a speaker from Brazil who's a couple of hours in a different time zones than us, but we're very glad that you can be here, that this is an appropriate time for you. So Pedro Bergamo is a pollination ecologist from Brazil. He did his PhD at the University of Campinas, and he's now a postdoctoral associate at the Botanical Gardens in Rio de Janeiro. And he'll tell you more about the topic that he'll go into, but it's something that a lot of us have an interest in this field, so we're looking forward to that. Just a couple of household things quickly. We would appreciate it that everybody please keep their sound and video off throughout the webinar so that we don't have those distractions. You're welcome to put your questions into the chat text box if you want to throughout the webinar, and then we'll look at them at the end of the presentation. But you can also raise your hand and ask questions at the end as well. Everybody is welcome to do that. And then in terms of SACNAS points, people that want SACNAS points for attending here, please just mention your name and a note about that in the chat box so that we can make a note of that as well. Right, I think that's all that we needed to say about that. Pedro, you may go ahead. We look forward to that. Thank you very much.
Great, thank you, Glynis. Thank you, Anina. And I want to thank beforehand the South African Association of Botanists for inviting me to present this talk. Can you all hear me well?
Yes, thank you.
Great, so I will then start my presentation about pollination ecology when there is no pollinators, not actually no pollinators, but in low pollination environments, when there is a low abundance of pollinators. So in this talk, I want you to keep your mind in patterns and processes that occur when many species are co-flowering, and not that only many plant species are co-flowering, but also many pollinator species are co-foraging. And then we are going now to see some patterns at this scale. So in the community scale, in multi-species communities. But of course, this is not a tropical image, as you can see. And then I will quickly move to the tropical environments. So here you can see part of Brazil, just to see, just to have a sense of from where I'm going to talk from, and from which environment I'm going to talk about. So I'm going to talk about this one that's called Campo de Altitude. So in a free translation, we can translate this to highland grasslands. You can see that it's this more light gray spots here that are surrounded by this whole matrix of Atlantic rainforests. So these are mountaintop grasslands that are surrounded by tropical forests. So once you are in a tropical forest, this is a picture from the Atlantic forest, and then you go up. Eventually, depending also on some of the the edaphic conditions, the soil conditions, the forest will eventually give up to ecosystem like this. So this is a picture from the from all of the data that I'm going to show were collected. This is the Parque Nacional do Itatiaia, or Itatiaia National Park. That is a park where the main ecosystem, one of the main ecosystems, is the Campus de Altitude, or this highland grassland. So you can see that the major vegetation components are grasses, so Poaceae and also Cyperaceae, but also we have represented many other herbs, mostly from the Asteraceae, Lamiaceae, Ericaceae, Melastomataceae families in this ecosystem. So here you can see another beautiful picture to get more a sense of how is this ecosystem. And here just one more beautiful picture, because I think it's always worth it to show beautiful pictures from the beautiful places that we are privileged to visit and study. So yes, I'm going to first show some natural history patterns of the plant-pollinator interactions there, so you can have more of a sense of what is the pollination ecology in this environment. So here you can see some of the animal pollinated plant species that we have there, and some of the pollination animals as well. Here in the first row, we have some species of large bees, mainly from the Chilocopine bombus genus. We also genera. We also have small bees. So here you can see some Asteraceae being visited by these large bees, a Solanaceae species. Here is a Scaloniaceae species. But we also have many flies from different fly families that are also pollinating animals there, like Tachinid flies, these large flies, but also many representatives of the syrphids that are these flies that also eat pollen. So here you can see flies visiting some Apiaceae species, but also here an Iridaceae species. And also, which are less representative in terms of visits and plant pollinator interactions, we also have some wasp species. Butterfly species are very rare as pollinators there. Ants also are very rare as pollinators there, and beetles. And also we have the only vertebrate animal that are pollinators in these highland grasslands are hummingbirds. We have 5 hummingbird species there, and then there you can see it is the same species that is visiting here a Bromeliaceae, and here a Lamiaceae species. And this figure summarizes all of the observations that we have made so far there. So this is just showing in terms of floral visits, so individual visits to individual flowers made by each of these groups of animals. So here you can see that native bees and flies are very important components. And as I have said before, wasps, beetles, ants, and hummingbirds are less representative in terms of visits. And here you can see the honeybee, that is an exotic species here in Brazil, Apis mellifera. That's probably, so we don't have much more data on this, but probably is causing a major impact in this ecosystem, because yes, you can see lots and lots of apis flying, visiting lots of different plant species as well. So here you can see all four photos are from apis visiting different plant species there, and you can see it visiting this Lamiaceae species, this Melastomataceae species, this Austromeriaceae species, and also visiting the Trifolium, that's also an exotic. So this is an interesting case of 2 exotic species from Brazil that have met again in another outside of their distribution, of their original distribution. So as I anticipated in the title of my presentation, we have low visitation rates. So this number means that if you sit on a plant and start to observe flowers, there is a chance that if it passes 1 hour, you won't see any visit at all. So you have less than 1 visit per flower per hour. So visits are rare because pollinator abundances are naturally low in this environment for most of these groups of animals. And here we have also classified the pollination systems for 60 and 63 plant species. Sorry. So here is showing how many, or this percentages are showing how many, the proportion of species, of plant species that are mainly pollinated by hummingbirds, or by bees, or by flies, or in combinations, or more even combinations of bees and flies, bees and hummingbirds. But here I have highlighted this group that comprises 1/3 of the plant species that are pollinated by diverse insects. So these are various combinations of at least 3 or more of these insect groups, mainly pollinated by bees and flies, but also by wasps, also by butterflies, also by beetles, and also by ants. So you can see that generalized pollination systems are much more common in this low pollination environment. So we can see that one of the, we are interpreting this as a response to the low abundance of pollinators. So the plants still rely on biotic pollination, but not, but using generalized strategies, maybe as a reproductive assurance strategy. And here you can see this other figure that are depicting this. Here in the right, the generalized plants that are most of these plants have smaller flowers. We have some endemic and some endangered species that were already evaluated there. You can see that most of these species are have yellow flowers or white flowers. And then you can see here in the left some of the more specialized plants in terms of the plant-pollinator interactions. You can see these plants are more present, generally have larger flowers and exhibit flowers like purple flowers, blue flowers, or more reddish flowers, pinkish flowers. And one last figure about these natural history patterns. You can see the flowering phenology here in the upper panel. These are all plant species aggregated together by month, just to see that there is a market peak in the summer months here in Brazil. So to December through February and March are when you can see, one can see more flowers or more species blooming. It corresponds to our summer, and here and in the summer in the region also. And here, the number of flowering individuals per plant species, just to see that most of the plant species are rare. So this is a very common pattern in tropical communities in general. But these are only represented by flower individuals or adult individuals or reproductive individuals. So we have some abundant plant species represented by reproductive individuals, but most of the plant species are very rare in the community. So now that hopefully you have more a sense of how are the main pollination patterns in this ecosystem, I want then to go to a more ecological, if you will, part of the presentation, when you can talk more about ecological patterns and processes. And I would talk mainly about processes that are mediated by density. And here, density, I'm saying specifically floral density. So we know that in most of the theory, and for most of the time throughout the development of ecological studies on pollination and plant-pollinator interactions, most of the theory expects that we will find positive dependence. What this means, that with increases in density, in floral density of a plant species, we expect to see more pollinator visits, and by consequence also more pollination rates, like more pollen reaching stigmas, a higher number of pollen tubes germinating on styles, a higher seed production, and so on. So this we can translate also to, I like to translate this because this makes it more clear, to advantage of abundance. So a plant species have an advantage in terms of pollination when it gets abundant. The consequences of this is that if pollination fosters and promotes abundant plants, these plants in turn can then compete with the rare plants. So we will expect that these plants will displace them, will exclude them from the community, or exclude them from the community. And then what you can expect is that pollination actually destabilizes coexistence in this local scale of local communities. So if everywhere else are equal, abundant plants would get more pollinated, would produce more seeds, the seeds would germinate more, and then we will gradually see all rare plants being excluded or extirpated from local communities. Of course, this is not what we see in nature. We see lots of rare species coexisting with abundant ones in tropical environments. So one of the conclusions of these early conclusions that maybe pollination is not an important process mediating this local coexistence, and local coexistence in local communities. But we wanted to investigate these density-mediated patterns in this environment that I already showed to us, to you, that is a low pollination environment. So we then established more than 100 plots, small plots, to measure these floral neighborhoods. So here you can see two species that are very close together. So this we established it as one floral neighborhood. This another floral neighborhood that is represented by 3 plant species flowering together, co-flowering. And then in these plots we measured in focal plants the visitation rates, the pollen receipt, so the number of pollen grains on stigmas and the number of pollen tubes on styles, and also pollen removal from anthers as a first approximation of female components of pollination and male components of pollination. We know that this is only the beginning of these pathways, but because we wanted to do this for a very large set of plant species, so we measured 67 focal plant species across these more than 100 plots. So we chose these first proxies for pollination, female and male pollination success. And then in each of these plots, we measure the conspecific density as the number of flowers of the focal species and individuals also from other individuals of the same species, and also the heterospecific density, that it means the number of flowers of other plant species, but only of this plant species that also shares pollinators with the focal one. And then we also have this measure that's here we are using this name of landscape, that is the conspecific density when you sum all of the flowers across the plots in 1 month. So we have this measure just to indicate if this is a rare or an abundant plant in the community at that point of time, because we wanted to separate the patterns from rare and between rare and abundant plants. So here, actually, when we saw the relationship between this landscape specific density, in other words, if a plant species is rare or abundant at the community at that point of time, we also saw that we actually saw that when the most abundant these plant species were at the community at that point of time, we saw less pollination or lower pollination rates. So we see less, so we see a negative relationship with pollen receipt and also a negative relationship with pollen tubes. Here, all dots represent a plant, a focal plant, and the distinct shades of gray represent if these individuals are from a plant. These individuals were surrounded by few or few heterospecifics. These are the black dots, or the lightest gray if these individuals were surrounded by several heterospecifics that shares pollinators with them. So you can see that these relationships, these negative relationships, were most accentuated if that focal plant was surrounded by many heterospecifics. So this also called our attention that maybe these plant-plant interactions, these interspecific plant-plant interactions, are also interfering in these density mediated patterns. And here you can see this in these graphics, because now our responsive variables that are the pollen receipt on stigmas and the pollen number of pollen tubes on styles, these graphs are showing the relationship between these pollination variables with the heterospecific density. So again, each dot here is a focal plant individual on one of those floral neighborhoods. But now here the shades of gray and black, they show if if that focal individual belongs to a plant species that is rare at the landscape scale or in the community at that point of time, or here in the lightest gray, if that focal plant belongs to a plant species that were abundant at the community at that point of time. So here you can see that heterospecifics most have a positive effect on the pollination rates of these focal individuals. And this positive effect were most accentuated for rare plants. Here the black line showed by the black line and the black dots. So this pattern that we interpreted as interspecific facilitation were most accentuated for rare plants. So rare plants in the rare species in this environment are benefiting more from interaction or from sharing pollinators with other plants there. And also the abundant plants seems to be in a disadvantage, and we are interpreting this because these abundant plants are more subjected to intra-specific competition. So they're competing more. The individuals are competing more between themselves when a plant species is abundant than when a plant species is rare. Here are also the same types of graphics, but now showing the relationships between pollen removal. So this measurement that we're using as a first proxy for male pollination success, and also the conspecific density at this landscape scale. So here you can interpret it that we are comparing rare and abundant plants, but also depending on how much these focal individuals were surrounded by these heterospecifics that share pollinators with them. And you can see here again positive relationships indicating also interspecific facilitation, but here it's interesting that we saw this hump-shaped pattern when abundant plants seems to be more on disadvantage, but also rare plants here in terms of pollen removal are also on disadvantage. And also here we also wanted to not just see these overall patterns for the whole community, but we also want to see these patterns depending on plant traits related to the pollination ecology. So here each of these graphs are showing the number of plant species that in blue are facilitated by or showed positive responses to the presence of heterospecifics. In gray, no relationship, and in red, the number of plant species that showed negative responses to the density of heterospecifics surrounding them. So you can see that the generalized plant species were most engaging on facilitative interactions, while the specialized plant species were engaging on both competitive and facilitative interactions in similar frequencies. Those plant species with large floral displays, this means that plant individuals display a higher number of flowers, were also engaging more on facilitative interactions, and those with small displays were also engaging more evenly in facilitative and competitive interactions. So summarizing these results, we saw most advantages of rarity in this ecosystem, and then we also interpreted these patterns as the opposite of what most of the theory were pointing, that actually pollination stabilizes coexistence, because pollination can be a force regulating the abundant plant species, and also here I use this word, but rescuing rare plant species or maintaining rare plant species in this scale of local communities. So we also wanted to see, after this study, we also investigated these floral structure patterns. So these, from those that are working with the community assembly theory. So this is placed on this theory, where you have like a community pool, and then if you have more competition, one can expect more flowers, more species with flowers that will be more different between each other. So a high trait diversity and a pattern of functional evenness, because then this will facilitate a partition of the pollination niche and the coexistence of these plants under competition. But under facilitation, we can expect the opposite scenario of a low trait diversity, because we then expect that sharing pollinators, that sharing pollinators actually benefits that plant species. So then we can expect a low trait diversity, representing that these species belong to the same pollination niche, and here a pattern of functional clustering. I don't know what happened here. Let's see if, no, this, sorry. Let's see if I can. Clean this. Oh, here, great, sorry. And then in this, so we have done some floral structure analysis. I'm not going into the detail of this, but the important message of this graphic is that here you can see the floral trait diversity. So when this gray triangle represents the observed value of floral trait diversity for this particular community, only for the plant species that were flowering in October of 2016, on November, and then going through May of 2018. So here the gray values are the observed values, and these empty circles represent the new values under some reshuffling of the trait values of these species. So if you see that if this gray triangle is much lower than this empty gray circle, then this is an evidence of this functional clustering. In other words, that the plant species flowering in this particular month are more similar than each other in their floral traits than expected by chance. And we could see also the opposite pattern. If the gray triangles were here, exhibited higher values than the empty circles, but we haven't seen this in any month. And in these months where most plants are flowering, so this that also coincides with the flowering peak of the plant community, we see these functional clustering patterns, clustered patterns. See, in other words, plant species that are co-flowering in these months are more similar than each other than expected by chance. And we also wanted to see if we also related some of this these measurements of pollination success to with the flowering synchrony of each plant species. So here in these three graphics, you can see each dot is a plant species actually, and here the flowering synchrony is a measurement of how much synchronous that plant species is to the rest of the plant community. And here, in each of the response variables here, in each of these panels is one of those pollination success variables. Because here we wanted to investigate if these plants are co-flowering with the rest of the community, if these plants are actually benefiting. So this is another line of evidence of this competitive versus facilitative interactions in this community. So for pollen removal, we saw that actually the most pronounced pattern was that plants with small floral displays, actually, if they were high synchronous with the rest of the community, they showed less rates of pollen removal, and the opposite scenarios for the female pollination components, for pollen receipt as well as for pollen tube number. So here you can see that there are some opposing processes, or at least it seems that opposing processes are mediating each of these pollination success components. So we saw some consistency between theory and observed patterns, because of course we were expecting facilitative patterns in this community. But when we compare male and female pollination components, we can see different stories, which is very interesting, at least I think. And this, so all these results are from this one particular community that I'm also always emphasizing, that is a low pollination environment, and we're always going back to this pattern to kind of explain what we are seeing in this community. But these patterns of negative density dependence or negative frequency dependence, and this role of interspecific facilitation through pollinators in maintaining rare species in plant communities is also are also being evidenced in other studies. So here, including one that was conducted in the Fynbos, here by Gita Benadi and Anton Paul, but also in one study conducted in the serpentine sips in the United States, that I'm not completely sure, but I think I can say that these are not actually low pollination environments. So maybe this the contribution of pollination to plant coexistence and to the maintenance of rare species is more widespread than what we could anticipate by theory. But of course, we need more communities being evaluated to get more a sense of this. And here I will show some final results. These results are, so this is mainly results from the PhD of Natalia Sterher that were also studying the same community with the same plant species, the same plots, so the same focal plants. But then she was looking at heterospecific pollen transfer. So all of the patterns that I showed to you was based on conspecific pollen. So I countings of only the conspecific pollen on stigmas. And Nati were counting and analyzing and the heterospecific pollen on stigmas. So here's some general patterns. You can see that species here in the responsive variables are the number of heterospecific pollen grains, the average number of heterospecific pollen grains per plant species. So each dot here is a plant species. So you see that those plant species with stigmas more exposed were those receiving more heterospecific pollen. This is very expected pattern. But this here was unexpected because those plants with specialized pollination systems actually were those that received more heterospecific pollen in comparison with plant species with the generalized pollination systems. Here we think that these are mostly because of the hummingbird pollinated plants, because hummingbirds have very indiscriminated forage boats. They exhibit much less floral constancy than bees, for instance. So they can, I think that this is reflecting actually hummingbird pollinated plants. And here Nati also have looked at these relationships with density. So here you can see that the proportion of heterospecific pollen on the stigma of that focal individual in that plot in the floral neighborhood. So each dot here is a focal plant individual. You can see that for those plants that are rare at the landscape level or with intermediated density at the low landscape level, in other words, that in that particular point of time in the community were rare or showed intermediated density, there was no relationship between the proportion of heterospecific pollen and the density of heterospecifics in the floral neighborhood. But for those abundant plants, yes, she saw this positive relationship. So those abundant plants are also more at risk of receiving heterospecific pollen, especially if they are surrounded by many heterospecific individuals. We're interpreting this that maybe rare plants have those mechanisms of avoidance, of avoiding receiving heterospecific pollen, but this is only speculative. We don't have further data on this. And this also to show that heterospecific pollen do have negative effects on the plant species. So here is the pollen tube success. And here are measures, these points are aggregated measurements of all those samples that show it 2 or more heterospecific pollen types on the stigmas, just 1 heterospecific pollen type, or no heterospecific pollen at all on the stigmas. And you see that those focal individuals with no heterospecific pollen had more rates of pollen tube germination, especially in comparison with those that received 2 or more heterospecific pollen types. So this is also aggregated for across all plant species in the community. So to wrap up, the pollination ecology here without pollination it's not really without, but in low pollination environments. In this particular one, we saw great high levels of generalization, patterns that point to advantage of rarity, several functional patterns that are consistent with what is expected under facilitation, but also some conflicts and opposing processes seems to mediate each male versus female pollination components. And that we are also speculating here that heterospecific pollen can be also as an additional potential regulation mechanism of those abundant plant species, also contributing to these advantages of rarity that we see that we are seeing in this environment. Of course, all these that I have showed to you are from observational data, community level observational data, the plant-pollinator interactions, plant reproductive success, the functional structure. And in the next steps, we now want to conduct some manipulative experiments. Oops, sorry, to confirm and to get more insight of the actual mechanisms behind generating all these patterns. So here, just to end, another beautiful picture of this beautiful place. I hope that I could show a little bit of the amazing patterns that we are seeing in this particular environment. And of course, I want to thank all the collaborators of all these the studies that the results come from. The Itatiaia National Park that harbors this marvelous ecosystem, and of course, everyone from the South African Association of Botanists for organizing this webinar, for inviting me here, especially to Anina, and thank you all for hearing me. Thank you.
Thank you so much, Pedro. That was great. That was very interesting. Thank you. I'm going to, where are the questions now? They have left me. Please add your questions in the chat if you want. Timo has a question. Please, Timo, go ahead.
Thanks, Anina. Hello, Pedro. Very nice to meet you in this context. And thanks a lot for your talk. I actually have 3 questions, and I don't know whether there's time to answer all of them, but I'm gonna ask them anyway. 2 of them are linked, and then the third one is quite different. So the first 2 questions are about your inference of low pollination environments. So I'm just curious whether you have any comparative data on whether the pollination rates that you observed in the community that you worked are particularly low compared to other areas in the world, and whether you find that a lot of the species in your community are pollen limited as potentially as a result of low visitation rates, and whether this differs between species that have specialized and generalized pollination systems. So those are my first 2 questions. Let me maybe stick with those. So for now, whether the rates are low comparatively, and whether this has an effect on pollen limitation, and whether that pollen limitation varies between specialized and generalized species.
Great. Yes. So the first question. So thank you, Timo, for your questions. And the first question, yes, there is a recent paper by Herrera, I think, that has a table showing like visitation rates per communities around the world. So there's no, I think it's like 5 or 6 communities, so not that plenty of data. And yes, the campus de altitude, the highland grasslands, have much lower visitation rates than all of those 5 or 6 communities that we have this data, or that at least that we have this aggregated data easy in a table. So yes, I think it would be very important to actually have more of this data available somehow. So we can have more this sense of what is a low pollination environment, what is a high pollination environment, because we are always saying that this is important, right? But we actually don't know if there are these gradients of pollination environments and so on. And the pollen limitation question. So we didn't conduct it like the pollen limitation experiments, like isolating flowers. We are starting to looking now to proxy of pollen limitation, that is the number of pollen tubes in relation to the number of ovules. And so this is very general. The only thing that I can say to you right now is that we have less pollen grains and less pollen tubes than ovules in the community. So in general, the community is pollen limited overall. we hadn't started to look at patterns per species or per groups of species yet.
Thank you, and thank you, Timo, for
Thanks
the
a lot.
question. I think we'll just go on to Steve's question, and then if there's time, we can go back to you, Timo.
Hi there, Pedro. Video. My question was actually very similar to Timo's, so this is perhaps more by way of discussion about the visitation rates. I was recently trying to find data on visitation rates kind of globally and found that there wasn't very much. So I'd be quite interested to know the review that you talk about. But interestingly, the one paper I did find, which was Thielen Ashman and Dan Schoen, had a visitation rate of about 1 visit per flower per hour for a meadow in North America, which is actually very similar to the value that you have in Brazil. So at least in that comparison, it's perhaps normal. And I must say that from my experience in South Africa, if I was getting 1 visit per flower per hour, I would consider that a very well visited plant species. I would consider that quite a high visitation rate. I know that certainly in most of the Cape Fynbos, the visitation rates are sometimes 1 visit per flower per week, if you're lucky. So yes, I think Timo's questions is this very much along the lines I was asking, which is really, is that really a low pollination environment?
Yeah, yes, I also am saying by experience of being there and observing there. So besides this aggregated number, it's also important to see the distribution between plant species, because this number that I showed to you, like 0.8, is very due to a few plant species. That these ones are indeed very highly visited, but most of the plant species, then you can also expect like this 1 visit per week. So yes,
that would make sense. Yeah.
Besides the aggregated number, it's also important to look at this distribution between plant species. Yes.
So that was also an average rather than a median, perhaps. Yeah. Okay, interesting. Thank you.
Thank you very much. Timo, do you want to give your other question?
So I'm actually going to hijack Timo's question because he's already had 2 and I'm using the same computer. So this is Ruth. Pedro, can I ask how confident you are of heterospecific versus conspecific pollen identity? Because having worked, I mean, there's so many species out there. We know that closely related species have got very similar pollen. It looks like a very big challenge to have processed the amount of data that you have to get these answers.
Yes, this is very tricky. I was not the one identifying the heterospecific pollen grains. So this was Nati's work. And yes, she had quite some difficulties also, because we had in this community, in the animal pollinated plant community, we have lots of Asteraceae, and Asteraceae pollen can be very similar, across species or genera. But she reached a level of confidence that at least that's not from the same species, or at least not from the same genera, but in most of the times, at least not from the same species, because we are also crossing this data with the flowering phenology data, and also the pollinator data. So if the plant species shares pollinators or not. So yes, we try to cross this information also to get more confident about what is conspecific and what is heterospecific. But yeah, it's tricky.
the fact that you found the same patterns when you looked at pollen tubes and when you looked at pollen receipt, if you consider pollen tubes the closest to an indication of reproductive success that you could relate to pollen receipt, suggests you are getting it right. It just struck me as quite a challenge.
Yes. I haven't thought this way also. By thinking that the correspondence between conspecific pollen and pollen tubes also indicates this. Yes, great. Thanks.
Thank you so much for that question. Glynis, please go ahead.
Thanks, Pedro, for a fascinating talk. Really interesting. I just wondered, in tracing, also quite technical, but in counting the pollen tubes, were you able to see if they actually reached the ovules, or was that not an issue? And if not, would closely related pollen grains be able to germinate on, for example, generalists or specialists? I wonder, is there anything known about that?
Yeah, so the first idea was to count the pollen tubes reaching the ovaries, reaching the ovules. But this was proven to be very hard for many species, due to methodological issues actually of staining the ovaries for many species there, because many species there have very protected ovaries, and so it was hard to stain the ovary to see the pollen tubes reaching the ovules for many species. So to have at least one first approximation of this reproductive success for as many as species that we could, then we only counted pollen tubes on the styles. And yes, there is potentially this error of counting pollen tubes that are from closely related species that could germinate, and could also germinate until the end of because we counted at the end of the style, but it can happen. A close related species could also, the pollen tube of close related species could also grow until the end of the style. But yeah, we just assume it that most of the pollen tubes should be of the same of that particular plant species. But maybe there is an error there also.
Thank you. Thanks.
Thank you.
Thank you. Yes, Timo, you may go ahead. Thanks.
Thanks. So this is my third question, and it's also relating to the graph that you showed from the PhD student on heterospecific pollen, specifically about the number of heterospecific pollen types and the response variable. And I want to ask, do you know whether that effect is due to stigma clogging? In other words, if there are more types of heterospecific pollen, there's less space for conspecific pollen, or is it some kind of interference that if you have multiple heterospecific pollen types on a stigma, there is less potential for conspecific pollen tubes to grow into the stigma? Yeah,
so the response variable is heterospecific. The proportion of heterospecific pollen in relation to the total pollen received. So maybe this could but actually this, yeah. So my most sincere answer is we don't know, actually, because it could be either stigma clogging or interference.
So I guess if you have the number of conspecific, the absolute number of pollen grains, both heterospecific and conspecific, you can actually evaluate those two options, which I think would be quite interesting.
Yes, I know that in these models that Nati was already accounting for the number of conspecific pollen. because of course you can see more pollen tubes because you have more conspecific pollen. So these were already accounted on the model. So it's like statistically separating only the effect of heterospecific pollen, but still I don't know, I'm not quite sure how to separate if it's sigma clogging or interference.
I guess with stigma clogging, you would expect that if the absolute number of heterospecific pollen grains increases, the absolute number of conspecific pollen grains would decrease. Whereas if it's interference, I guess the effect would be due to the number of heterospecific pollen grains, regardless of the number of conspecific pollen grains. Possibly.
Yes. So yes, if I can remember that well, it's the second option. Yes. So the heterospecific pollen, the negative effects of heterospecific pollen are there regardless of the number of conspecific pollen. So yeah.
Okay. Thanks. That's very interesting.
Great. Thank you very much for those questions. I'm just checking in the chat box quickly. I don't see any other questions. Is there anybody else that has any questions? Right. Hope I'm not missing anybody, but I don't see anything. And I think it's all in good time now. So it's just about an hour since we started. So I just want to thank Pedro. Thank you. That was really great and interesting. And we look forward to your experiments and those results. I'm sure that's going to be interesting as well. And I hope that you can come and visit South Africa next year. We hopefully will have the pollination symposium in Cape Town. So you must see if you can make a plan to get here. Thank you, everybody, for joining in and listening. We appreciate your time. And our next webinar will be in October. So look out for those adverts as well. Thank you very much, Pedro.
Thank you. Thank you again for inviting me. Yes, and now I'm very looking forward to go to South Africa. So
yeah.
Yes, it would be great.
Great. Great. Thank you very much. We will have a recording of this presentation as well. So you can let me know if you need access to that.